Welcome to our RNA-Seq Alignment and Analysis webinar. My name is Katie Maxfield, and I'm hosting the webinar today. I'm here with Farhan Qureshi, who is part of our sales team, and he's also very familiar with our RNA-Seq workflow. Originally, we had planned for Matt Kaiser to give this presentation, but he had an emergency he needed to attend to, so Farhan has graciously agreed to step in and fill his shoes. You may notice that your phone has been muted. Please feel free to ask questions during the presentation via chat. To do that, just send a chat message to host, that's me, and I will pose those questions to Farhan throughout the webinar. And we'll try to get to as many questions as possible during the hour, but any questions we don't get to, we will follow up with you after the webinar. So with that, I am going to turn it over to Farhan.
Thank you, Katie. Well, I thank you for joining today's webinar presentation for the RNA-Seq webinar workflow. We recently introduced Lasergene 10.1 last month, which integrates our analysis and alignment tools in one package with easy-to-use workflows that you can go through without changing any higher-level parameters like most other software tools. So today's agenda is to go over the RNA-Seq workflow. I'll go into the actual software, walk you through the project setup, then I will go into the alignment, visualization, and analysis tools, and then I will go into our gene expression analysis tools. And as Katie mentioned, if you have any questions, you can just chat her, and she'll interrupt me, and I'll be able to answer them for you. And with that, I will jump right into the software. So I will start off with our DNASTAR Navigator. The DNASTAR Navigator was introduced a few months ago with version 9, I believe, last year. And with it, we added an integrated search that you can search through all of our software programs for any workflow question you may have. And in addition, it separates our packages by different suites. So we have the DNA sequence analysis section, if you're doing DNA sequence analysis. We have the next generation sequence assembly analysis, which we are doing today with RNA sequencing. So I have this expanded. But you can expand any of these other ones to focus on those specific modules. And notice if you hover over any one of these programs, you will get a quick summary of what the actual program does. And then you will also see tutorial video links right under it that you can click on and go directly to the tutorial video for a specific workflow. We have over 100 videos for various workflows in our software. To view a full list of those videos, you can simply click on More Videos and go directly to that section. If you have a more specific question, you can click on this I button and click on then Search. And then in the search, you can type in any term that you want. We'll type in RNA-Seq, for example. And it will go through the whole software documentation and look for any terms that match your criteria. And here we found RNA-Seq. You get an explanation of RNA-Seq in our software, and we have other listed in here as well, such as normalization and other search terms. And so we'll close out. And this is true for all programs. You don't have to specify a specific program. It'll do it for the entire suite. So for the initial alignment of our RNA sequencing data, we will open SeqMan-NGen. SeqMan-NGen has a workflow-based interface that is easy to use. It will walk you through your project setup so you don't make any mistakes. And as you're making changes or picking your workflows, the parameters in the background are actually adjusting to the selections you're making. The parameters have been optimized for your specific project type. So this takes away from all the complexity of adjusting manual parameters or not knowing what to do or running the risk of having an alignment that doesn't look very good because of unknown parameters. So that factor we take out of out of your hands. We take care of that through extensive testing of our software to make sure for each project type the parameters specified are correct and produce best results. So start off, you will see this welcome page. And in this welcome page, you can start a new project, or you can load in an older project if you want to rerun. And here we'll just continue and start a new assembly file. And below you will see the navigation buttons and help. If you ever have a question as to what a specific workflow is, or a question on a parameter, you can always click on the help on that specific page, and it will take you directly to the help menu for that section. So for help here, it will take us to the welcome page, help documentation. And now we'll go next. And this is where you specify your project type. As you can see, I have selected here transcriptome for our RNA-Seq alignment. But in addition to RNA-Seq, we also do other workflows such as genome assembly, targeted resequencing, exomes, and metagenomics. So the software is very flexible for different project types. Today we'll focus on RNA-Seq. And once we've made that selection, we'll click on Next. And we will leave the default options here for templated assembly, since we are using a reference sequence. And then in project setup, this is where you specify your project name, the save location for your output file, and the temporary file location. If you're not sure that your computer meets the minimum requirements for a specific workflow, you can always click on our technical requirements link, and that will take you directly to our web page. It opened here. That breaks down the technical requirements top of the page. This will break down the technical requirements based on the various project types and organisms that you may be working with, or that are very similar in size. For example, we have the human exome genome, the rice genome, and yeast genome. And based on your project size or the size of the organism, you can pick and choose which hardware requirements would be needed for your workflow. And we have direct links for both Apple and Dell with those requirements pre-specified on our web page. And we'll go back to the project setup. And once we have specified the project name and location, we can go to the next page where we can specify our template file. You have a couple options to input a template file. You can either download one from ncbi for your specific organism, or you could import your own file that you have saved locally. But you can also import a genome package that we provide on our website. And here's a link to the genome package. The genome package includes the reference file from NCBI, as well as the dbSNP database, the COSMID database, as well as GURP score database into one package, so that during your alignment, as your reads are being aligned to the reference genome, all of these databases are being incorporated into the alignment, and in the output file you will see the dbSNP IDs for the annotated SNPs. For example, you will be able to see the GURP scores for highly conserved regions across all species, or COSMIC data for cancer-identified genes. So all of the common organisms that are asked are on this page for the different versions. Human, for example, we have 3 different versions, with the 135 build being the latest one. We also have mouse and rat and some of the other ones. And if there is a genome package you would like us to build for you that's not on this page, you can always send us an email or click on this link down here. And I have loaded in the human genome data package, the genome template file from our website. And now I will go to the next page. And this is where I can specify my reads. Our software is compatible with all of the major sequencing platforms. We have Illumina short read, long read, 454, Ion Torrent, PacBio, or other. The data that I'm working with today is Illumina short read paired end. It's for 6 tissue types from human across various stages. And as you can see at the bottom of the screen, I have paired end data loaded. For each sample, notice there are 2 files. So if I wanted to pool both files, both lanes into one sample, I would simply click on this multiplex data. By default, it's turned off. You can click it to turn on. And once you've selected that, you can go into Customize Sample Names. And here you can see the multiple lanes. And if you assign a single name to 2 or more files, those files will be pooled as one experiment. So these 2, for example, will be just one experiment for brain, and it will just show up as brain in the final output file. Similarly, for the other tissue types, breast, heart, liver, testes, and UHR, we have 2 of these each pooled into one experiment. And once you've specified your names, you can click OK. And now we will go to the next screen. And this is where we have the assembly options. And the parameters that are filled in here are the default parameters that were added as you were making your selection. So for the Illumina data set, we have the MER size set to 19, the minimum match percentage to 93. The genome ploidy for the human genome we have deployed. These parameters will change based on the read technology you choose and the workflow you select automatically to best meet your needs. However, if your alignment does require some fine-tuning, you can go into these advanced assembly options and use some of the fine-tuning parameters that we have available. You can always contact us if you're not sure what parameters to change, or if your alignment is not as good as you want it to be. We have support available for you. And once you've gone through the project setup wizard, specified all the parameters, if you made any changes, you can click on Next to see a summary of all the selections you've made. This is a text file that will be saved, and it shows everything that you've selected, the reference file, for example, the reads that were imported, the pair distance, if that information is available, and the group name that you've assigned to it. So all of the options that you've selected and changes that you've made will be reflected here, as you can see. And at the very bottom of this page, you will get a list of all of the parameters that are being taken into consideration for this alignment. You can save this and import this later on if you wanted to fine-tune your alignment. You can do that and just go into the advanced options in order to change some parameters, but you don't have to run through the project setup from start. Once you have set up your project and you're ready to go, you click on Assemble. And I won't do that right now in the interest of time. I have assembled this already. And once the assembly is complete, or the alignment is complete, SeqMan Pro will open. SeqMan Pro is the analysis tool for the aligned data. And initially, you will see this window. This is a project summary window. On top, we will have a list of all of the contigs, and each contig will represent a single chromosome.
Farhan, sorry to interrupt. Before we get into the SeqMan application in detail, we had a couple questions about SeqMan Engine. One was, at the very beginning, there was a temporary file location specified. Do those files stay on the hard drive after assembly, or is that just a truly a temporary location? And what happens to those files after the assembly?
The files in this location are used during the alignment of your data, and they do remain after the alignment. You can delete them if you want. However, if you anticipate rerunning your alignment, you can leave that in there, and the alignment speed will increase significantly. But if you don't have the need to rerun your alignment multiple times, then you can definitely delete those files. It's just
Great.
temporary space.
Okay, thank you.
All right, we'll move back into SeqMan if there are no additional questions. So when SeqMan Pro opens, we will see this config summary window. On the top, as I mentioned, you'll see a list of all the contigs, and each config will represent a chromosome. In this project, I only have human chromosome 1. I did not align to all of the regions in the interest of time. And the next part would be to go into the different views that we have to offer to do the analyses. So we have this alignment view to look at the reads, the strategy view to look at histograms and coverage. And we'll go into alignment view first. So by default, this is the alignment view. We have the full human chromosome 1, total length on the top right corner. We have the position information on the top left corner for the spot we're on. And right next to it, if you click on a specific location, you'll also get the depth at that specific position. On the left, we have quick shortcut buttons for zooming in, zooming out, or viewing sequence name and other information. And on the top, we have the file options to switch between the different windows. Most frequently, you will just use this config option and switch between the strategy view and the alignment view. In the middle part of this alignment window, you will see this gap that has features. So each of these lines that you see will represent a feature. And if you hover over any one of these, you'll get a full summary of what the feature does and where it came from. This, for example, is TTC24, and it gives you the coverage and other information that is derived from the reference file. And on the bottom, you will actually get the reads that go into the alignment. And as you can see, we have this these yellow sequence, and for each sample, right here, this position, we have only 2. Let's go into deeper. So here, for example, we have brain, heart, liver, testes, and UHR, 6 different sample types, and the yellow sequence is just a pseudo consensus. If you expand any one of these by clicking on this triangle, it will show you the actual reads at that specific location for your sample, and you can do this for every single one. So as you can see, it's very powerful, very smooth and easy to use on a desktop computer, full human chromosome 1. And this is just a smaller project. You can, like I mentioned earlier, have all human chromosomes in one project. Okay. So that's just a quick overview of the alignment view. Now we will go into the strategy view, and to do that, you can simply go into the config menu and choose strategy view. And I will maximize this as well. And the layout of this view is very similar to the alignment view. On the top, you have the file options. On the top left, you have the quick shortcut keys, buttons for zooming in, zooming out, and viewing sequence information. Then on the top left, we have the positional information, and then we have a scale right in the middle section, as you can see. You can scroll throughout the bottom And we have the coverage threshold histogram right under the scale. We have the depth of coverage graph, pair consistency graph, and then we also have the feature arrows, just like in the alignment view. So very similar. You can make this feature view larger or shorter depending on the number of features that are at that specific location. So we'll leave it right here. If you hover over any one of these histograms, for example, it will explain to you what the different coloring means. And at the bottom, we have the actual reads that are aligned at that specific location. And you can, as you can see, scroll through. And you can look at peaks and different coverage regions. So very quick. Looking at the full human chromosome 1, and that's the total sequence length. So that was a very quick overview of the two main window of views in SeqMen Pro, alignment view and strategy view. Now we will go into some of the other tables. First, I would like to show you our SNP report. And I'll have the alignment view open at the same time so you can look at the SNPs. So to go into the SNP report, you click on the SNP menu option and choose SNP report. I'll move these around. And in the SNP report, at the very top, you have all the filtering options. And in the top left, you'll see a number. This is a live number that shows the total number of SNPs reflecting the filtering that we've added. So for example, right now we have about 2,100 SNPs. But if we change any of these filtering options, such as we can switch between annotated SNPs that will only show SNPs that are in the reference sequence annotated or from the dbSNP database, or we can choose to view novel SNPs only, or we can view both. If we say novel SNPs, we can see this number has been updated to about 1,300 SNPs from 2,100 previously. We'll show all. You can decide to filter based on percentage. Right now it's set at 50%, which means at a specific position, there are at least 50% of the bases are SNPs. So right here, we can up this to 75%, and we're down to 1,800 SNPs. We can choose to view SNPs that are within a specific distance of a coding region. It's 200 right now. You can say 100, and the numbers will increase and reflect that. And then you can also use some of these other filtering tools, such as showing coding SNPs only, or SNPs that cause coding change or nonsense changes. And then we have the different columns. Here's the MID column, which has the sample names. We have some of the contig IDs, the type. It differentiates whether it's SNP or an indel. Some of the more interesting ones is splice. With this splice, it will tell you whether there are exonic regions that were detected. And if I click here, for example, it will jump to that specific location in the alignment report because all of the views are interactive with each other. And then you can look into more detail in this view. And we can also look at the impact column, which is very interesting. It will inform you whether it's a synonymous or non-synonymous change, or whether it's a frame shift, and it will color code it. So this one, for example, it detected as a frame shift, and it's spliced in the CDS feature. And we can see the CDS feature over here, and then you can look at the actual indel that was detected over here, down in the alignment view. And we have other statistical information as well if you scroll to the right. So as you can see, the SNP table is very in-depth. All of these columns are sortable, and you can move them around depending on the SNPs that you might be interested in. So that was a quick overview of the SNP table. We will now move into a different view. I'll open the strategy view back up again. And then I will also open a feature table. For RNA expression analysis, this might be more relevant. In the feature table, you can sort based on feature type, genes, CDSs, mRNAs, and then we have a list of names as well that you can use and locations. And then we have other information as well, such as SNP count and depth of coverage if you scroll to the right. We have this ATP A2B2 gene selected. If I click here, it will jump to that specific location in both the strategy view and the alignment view. So as you can see there, they're all interactive. And here is the ATP gene that we selected from the feature table. And all these windows are resizable. So you can look at more sequence data. I'll collapse these so that it's a little bit easier to see the sample names in the alignment view. So you can scroll. And in the strategy view, you can see the histograms and the coverage at that specific location. I will actually now maximize the strategy view so I can focus on some of the exonic regions. So the gene that we selected, I'll zoom into it just a little bit more. Okay, so as you can see, this is the gene that we selected, and we have peaks all over, as well as we have paired consistency peaks. These reads that are aligned at these specific regions, they are exonic regions. And if you zoom in, each one of these arrows with coverage is representing an exon, and you can see the reads are aligned to that specific region. If you look more closely, we have these reads that are aligning right here, forward direction, and we have the reverse direction, and you can do that for other regions as well. You can click on any position and jump right to it in the alignment view by double-clicking, or you could just stay in this specific view. And as you scroll to your right, you can look at all the sequence names and the read information. I'm going to hide these sequence names so it's a little bit quicker that way, so you can get a better picture. Here's a quick summary of all the reads that are at that specific location. I currently have the settings set to show only the reads that are split at exonic regions. And these are the places where you'll most likely see the splicing occur as well. So here's a very good example. At each peak, we have very good coverage, as you can see. And you can do that for any gene.
Farhan, we've got lots of questions coming in about SeqMan Pro. If this is a good time, we can do a few of them now and maybe come back to some at the end.
Sure.
So one question is, you showed the SNP report. Is it possible to save that report to a tab delimited file, for example?
Definitely. Most of our reports are exportable. All you have to do is simply open that specific report, then go into File menu and choose Save SNP Report, for example, and it will give you the option to save as a text tab-separated file, which you can then use any other program or viewer.
Great. And then another question about SNPs. You talked about using the filter to show only annotated SNPs. Is that only available if you use the genome template package, or in what cases do you have that option?
It's available when your reference sequence has an annotated SNP in it, or if you use the reference package that has a dbSNP database. If you don't have a dbSNP database, then your reference sequence must have an annotated SNP. A lot of the sequences you can download from ncbi have annotations for some of the more common SNPs, and those would appear in the list as well. But if you don't have any, you can create your own annotated reference sequence using some of the tools that we have in our software. And for more specific details, we can set up another webinar or answer emails and show you how to do that.
Great. And then a final question here. In this particular example, you've aligned to a single chromosome. A question came in, is there any difference in quality or perhaps bias of the aligner if you use a single chromosome versus using the entire genome?
There's not. You can use the entire genome, and the aligner is efficient enough to determine, to remove the bias, and it will give you the same result whether you're aligning to one chromosome, like in this example, or to the entire genome. I actually had a different alignment that is for the entire human genome, which I did not open in the interest of time. It takes a few minutes to open up initially. But the results are the same, and there should be no differences.
Great, thanks. We have lots more questions, but I'm going to let you move on, and we'll try to come back to as many of these as possible.
All right. Thank you. So that was a very quick overview of SeqMen Pro for the alignment analysis of the RNA-Seq project. And now we will step into a different program, ArrayStar, to do our expression analysis. ArrayStar originally started off as a microarray program. Since then, it has evolved into an RNA-Seq analysis and a set analyzer. So to open ArrayStar, we go into the navigator and simply click on ArrayStar. And here we can import our RNA-Seq project that we aligned using SeqMen Engine. And to do that, simply click on RNA-Seq project. And then we can import our folder. And now you'll see a summary of all the tissue samples that were in that alignment. We have all 6: brain, breast, heart, liver, testes, and UHR. And then we go next. And this is a screen where you specify the normalization method that you wish to perform for quantification. By default, we have RPKM selected. However, you can choose some of these other methods as well, such as RPM, RPK, and some of the other ones we have listed. And finally, you will have to select the feature type you wish to perform the quantification against. By default, we have these listed, but you can use the pull-down menu and align, or not align, but quantify against a specific feature type of your interest. I did this for CDSs, so I will select this. And as you're making your selection, this number on the right will update for that specific feature type. For CDSs, we have about 3,300 features, 5.36 megabases, for example. For genes, that number will also update. And once you've made all these selections, you'll simply click on Next to begin the alignment. No, I'm sorry, not the alignment. I keep on saying that. I apologize. It's not the alignment. It's a quantification. The alignment has already been done in SeqMen Engine. So you click on Next, and once it's finished, you will get this experiments list view in RNA-STAR, and you'll have a list of all of your samples right here. And if you click on any one of these samples, you can get additional details on your right, such as the normalization method that was used and the total number of genes in the experiment. For more details, you can click on the pre-processing under experiment attributes, and you can look at actual statistics during the alignment of sequence engine as well as the quantification in RNA-STAR. So here you can see we quantified against CDS features, and you get more statistic information down here. And you can do that for each sample. Also notice on the bottom right corner, we have these quick action links, and these allow you to quickly jump to a different view or save a specific gene set of interest. So we'll switch to brain, and we'll choose to open the scatter plot. And in the scatter plot, you'll have two axes, X and Y. Both of these represent the expression values that were quantified using RPKM. And you can choose to compare different experiments. On the Y-axis, we have brain. And on the X, we'll choose heart. And this will update for your selections that you're making. You will also get quick statistical information on your right, such as R squared. You will also get some fold changes. And if you click on any one of these links, if two-fold change, for example, it will highlight those 14,000 genes that have two-fold change in these two experiments. You can also do the same for these other options, such as four-fold or eight-fold. We also have some additional advanced filtering tools that we'll get into in a few minutes, but this is just a quick overview to get you started in your analyses. Now, if you want to do a quick heat map hierarchical analysis, you can use some of these quick action links again on the bottom. Based on your selection, it will go through and generate a heat map. And from here, if you hover over any one of these genes on your right, you will get a summary of what you have currently selected if you click on it. And you'll also get a list of the neighboring genes above and below it. On the right, you will see a histogram with the color coding. The more red it is, the more highly expressed, and likewise, the bluer the color, it's less expressed. And for more details, you can look on the bottom right corner, and this is displaying details for the gene I have selected. It's a little bit faint and difficult to see, but it's this one right here, the PKIB. And you will see additional details such as the gene name and other information on here. And any of these links that you see, you can click on, and it will take you to that specific database for more information. It will take you to the original source. So that's the heat map. Now, in addition to these two views, we also have a gene table. The gene table gives a summary of all the genes in the project. And if you look at the bottom of this table, you can see we have 32,702 genes in this project. We have these columns on top. We have the name. And any of this information that's added will contain the annotations that are in the file, or the gene ontology that was downloaded, or expression values. So I have added here the expression values for all tissue samples in all genes. And to do that, you will just use the shortcut commands on top. These are uniform in all views. You will have shortcut commands for heat map, scatter plot, or any other view that you're in. And for example, if I wanted to add expression values in the gene table, I would just hover over the 1, 2, 3, and it will give me a summary of what this does. I can click and hold, and then I can choose to add expression values for all experiments, which I've done already, or I can add it to selective experiments. So here we have brain, breast, heart, liver. I can also add the raw count, the actual number of reads that were aligned at a specific position. The other information that you see here, product and Go ID, that is derived from this ABC pull-down menu. And all of the options that you see in here were taken from the reference file. So if any one of these is not in the reference file, you wouldn't see that option. And we also have the ability to import gene ontology information from the GOA consortium. And to do that, you'll simply go into the data table and then download annotations. I have that window open already, so I will go to that location. And here, you will see a live view of the database from the GOA consortium, Gene Ontology Consortium. And you can focus on, you can click on your specific organism and then download the gene ontology file for your organism, and then click on import, and it will bring it into the software. So this is not maintained by DNASTAR. It is strictly the Gene Ontology Consortium. It's a live window that will allow you to easily import a gene ontology. So once you've imported your gene ontology, we can go to the gene ontology view, and you will see by default the 3 processes: biological, cellular, and molecular. Now you can focus on specific workflows or pathways, and to do that, you can expand any one of these. So if I wanted to focus on biological processes, I can click on plus sign, or I can double-click on it. Notice by double-clicking on it, I'll see a progress bar, and there will be some statistics that are calculated, such as a p-value and the z-score. What the software is doing is going through all the genes and looking for those specific terms and calculating a p-value based on those terms. So if, for example, biological processes appears in all 100% of the genes, it will have a very high z-score and very low p-value. And you can further go down the list and focus on very specific pathways that you might be interested in. And once you've identified a pathway, you can right-click on any one of them and choose to show them in all views. So let's say we want to look at the cellular development process. I can click on here once, right-click, and say, select current cellular development process in genes in all views. And now if you switch to the scatter plot, for example, those genes will be highlighted in white. And from here, you can perform further analyses. You can then go into, you can perform a k-means clustering, for example, or you could go back to the gene table to get more information using these quick action links. Let's go into the gene table here. And you can get a quick summary of all the genes that were selected that met our criteria. You can see the expression values in each tissue samples and the annotation information. One thing I wanted to mention also is that in addition to the expression values and the annotations, you can also add fold change. And to do that, you would simply click on this 2-up button over here, and then you can select your control and the actual experiment. And that will add a new column in your gene table. And all these columns can be renamed, And to do that, you simply right-click and choose Rename Column. And we'll leave it as is for now. And let's go back into gene ontology. So from here, as I mentioned, you have the statistical information based on your criteria, but you can apply additional stats by using the quick settings over here. You can add a FDR p-value correction. You get a bar graph over here. You can get a pie chart. You get a list of p-values over here. So that's a very quick overview of the gene ontology view. Now, for some advanced filtering of your data, you can go into our filtering menu and then choose Filter All. And here you can set different criteria. By default, you'll have one row, but you can add as many conditions as you like. So in this example, we will choose fold change between brain and heart of 5 or higher in brain. And then we'll also add another criteria of expression. So let's add expression level in brain greater than 7. And we'll perform a search. And the search results will be listed in the bottom half of this window, where you will also see a total number of genes found that met your criteria. And if you scroll to the right, you'll also get the additional information that you can also view in the gene table. Now you can also save this search by clicking on the paper button over here with the asterisk. And when you click here, you can specify a name for your search, and the notes field will be pre-filled with the search criteria that you used, and that will save a lot of time. Likewise, you can also perform additional searches. Let's change this to second experiment and expression in heart. And now we have 123 genes, and you can search this as well. You can save it. And I've saved this beforehand, so I won't save it again. And then furthermore, you can add other criteria such as gene classification or ontology. And then you can further pick specific pathways that you might be interested in from the list. We have electron carrier activity. Decrease this. And here, for example, we have 4 genes that met these criteria. You can add or remove any one of these, as I mentioned earlier, depending on the criteria or the stringency that you want to set. So I have created a few gene sets here that I've saved. And now I will go into the gene set view. And to do that, I can simply close the filtering window and then go into Data, Show Set List. And here I have a list of all the gene sets that I created. As you can see, there are 4 different ones. And these I can use to compare amongst each other in our Venn diagram. And to go to our Venn diagram, I will simply go to Graph and Venn Diagrams. And here you will see by default, we have a dartboard. The dartboard is a very good analysis and visualization tool if you have more than 3 sets created. It's less clunky, and it's easier to kind of see where all the genes lie. And if you hover over any one of these sections or overlapping regions, under the details pane, you'll get a summary of all the genes that are in there. And I'll actually go back to the filtering window here, and we'll save this one as well. and we'll choose to open the set list at the same time. And it looks like I'm having some refresh issues here. But let's go into the Venn diagram. And here, as you can see, we have our set 5 listed in here as well. So this is a very quick overview of all the different views in RNA-STAR for RNA-Seq. It is very powerful, and if you would like to go more in depth about a specific feature or view that we could not cover today, please let us know, and we can set up another webinar especially for you, focusing on your specific workflow, or you could send us an email or call us directly. Now I'll go back into my slide here. So that was a conclusion of our RNA sequencing workflow. In short, our software is very easy and powerful to use. It's compatible with all platforms, sequencing platforms, Ion Torrent, Illumina, Roche 454, for example, all major operating systems, Mac, PC, as well as Linux. We have easy-to-use analysis and alignment tools. The software is very low cost. For less than $5,000, you can get the entire Genomics Suite, and technical support is provided on a personal basis with every purchase, and also before the purchase. If you have any questions, we can provide technical support during your demo or free trial. And with that, I thank you a lot for joining my webinar today, and I will hand it over to Katie again.
Great, thank you, Farhan. We do have a couple of questions here. One is, if you want to import an alignment from a different program other than SeqMan Engine, is it possible to do that using RNA-STAR with QSeq? And if so, what alignments are supported? RNA-STAR
QSeq, actually you would import it into SeqMan Engine first. You can import an alignment if you have a BAM file output. That is the only output that you can import. However, you would need to make sure the reference file that was used to generate that alignment is also used in SeqMan Engine, because even if there's a difference of one base pair, all of the downstream alignment will be invalid. So you must make sure that you have the same reference file. So yes, you can import an alignment from a different aligner. We don't recommend doing that. It's best if you use our own, but you have no other choice, and you do have access to both the BAM file and the reference, then it can be done.
Great, thank you. And a question that came up when you were showing SeqMan Pro, actually, but I think probably applies to RNA-STAR as well. If you're looking for a particular gene, can you search in those two applications to find that feature and jump right to that?
Definitely. In RNA-STAR, I will open RNA-STAR again.
Let me switch it back to you.
I just want to show you how easy it is. So in RNA-STAR, in the gene table, we have this search bar on the top right. You can type in any gene name. For example, if you type in TP73 and click on search, it will bring all those search terms to the very top, and it will also give you the expression values and other annotation information for that specific feature. So yes, it is searchable.
Great, thank you. I'm going to switch it back to my slide for just a moment. We do have some more questions, but we're running a little low on time, and I want to take care of a couple administrative things. First of all, as Farhan mentioned, we can do one-on-one webinars with people. So a lot of the questions that came in today were pretty detailed, and we'd love to get into those with you, maybe even looking at your own data, either via email or a one-on-one webinar. And as for this webinar, if you missed any part of today's session, there'll be a recording up on our website in the next day or two, and that's on our webinars page. And we'll also email everyone a link to that recording. So don't worry if you missed something or if you want to watch again, that will certainly be available. And then finally, another resource we have available is our videos page. Here we have a large collection of videos, over 100, and growing by 1 to 2 videos every week. And these are usually 3 to 5 minute videos showing specific workflows within the software, including some RNA-Seq and other next-gen workflows, gene expression analysis workflows, and also some traditional sequence analysis workflows. So if you want to see more of the software, that's another great place to start, and that's DNASTAR.com/videos. And lastly, if we didn't get to any of your questions, we will contact you via email, so don't worry. And if you come up with more questions after the event, you can email me at webinars@dnastar.com. And we'll also be holding some more webinars in the future. Our next webinar is scheduled for November, and that will be an overview of our Protein 3D application. So a little different topic than today, but we certainly hope you can join us for that as well. I thank you all for joining us today, and we hope to see you again soon.